About the 48 hours chick embryology. Today we'll be learning about the 48-hour of chick embryo. Before we begin with that, there are a few terms which we need to get cleared about. That was formation of notochord. Notogenesis, a narrow strip of plastodome just in front of the primitive knot, consists of presumptive notochordal cell responsible for the formation of notochords. Notochord is visible carnial to the Hensen's node and in between the neural fold. So there is a plate-like formation which takes place over the Hensen's node and near the neural fold, which will lie between the neural folds, the two neural folds. Notochord is rod-like structure which will be used for supporting the body structure of the animal. Then talking about the somites, which we have already seen in the 24-hour chick embryo, the somites are formed. These somites, they are the blocks of mesoderms. Okay, before the somites are formed, there is going to be a block of mesoderm which will be later formed into a somite. They appear on the either side of the notochord which is getting formed. They give rise to number of structures including skeletal muscles, bones, and the dermis of the skin of the animal. Formation of neural tube, that is neurogenesis, in front of the primitive streak where there is going to lie the neural plate. Okay, these cells fold and roll up and unite in the mid dorsal region enclosing the neural tube. So the neural groove will be formed and the neural tube will be formed later on. After the neural groove, the neural tube is getting enclosed in within it. This will later form the forebrain, midbrain, and the hindbrain region. When we were seeing the 33-hour chick embryo in our last lecture, we had observed the various structures which are formed. Okay, all the way from the 13 somites, we have also seen the structure of the brain, which was already divided into three parts. Okay, so in this picture also, you can see the brain vesicles, the optic vesicles, the heart, which is formed on the right-hand side portion. Okay, and you can also see how the primitive streak is actually on the posterior end, and all these new development is taking place anterior to the primitive streak. The heart formed on the posterior part of the brain vesicles, and you can see two vitelline veins are entering into the heart from the small blood islands which are formed on the outer periphery of the embryo, supplying blood to the heart. Optic vessels are formed that indicates the eye formation which is starting. The two optic vesicles emerge out from the brain region. Heart could be observed near the posterior of the hindbrain. Okay, you can see the pulsating heart if you see a video of this particular chick embryo. Now coming over to 48-hour chick embryo, this was what picture we saw, and we had also learned about the torsion which occurs in the anterior region, due to which the particular embryo gets a question mark shape of the face, or you can say the structure, the shape of the embryo becomes a question mark shape, or a small S-shaped embryo will be seen in the slide. Now talking about the 48 embryo, what are the various features of this embryo? There is appearance of cranial flexure, that is in the cranial region. You will see a flexure, a turning which is taking place, the bending of the embryo is taking place. That is the cranial flexure and the torsion. Appearance of 11 neuromeres can be seen in the picture. Three neuromeres are present in the procephalon region. Okay, the first brain region, which is called as procephalon, there you can see three neuromeres. Two are present in mesocephalon, and six neuromeres are present in the rhombencephalon region. That is the hindbrain region. Okay, so you can see this through the diagram which is given outside. Formation of secondary construction in the brain can be seen, that is, which is dividing the three brain portion. For completion of the vitelline membrane or the observed in this particular embryo, development of intra-embryonic blood vessels, capillaries are forming. You can see that formation of two pairs of aortic arches can be also seen in the image. You can see the first aortic arch over here, Okay, this is the first aortic arch which is formed here. You can see the atrium being formed over here. Okay, so the heart is starting to get formed properly and more accurately. Okay, you can see, as I told you, you see the first aortic arch, the second and the third aortic arches. So you can see how the arches are formed, twisting of the heart and the formation of chambers in the heart. Because since it is a avian animal, it will have a four chambered heart. So if you see it, you will see that the restriction constriction is taking place and the heart formation is taking place. So the formation of two pairs of aortic arches and then this twisting of the heart can be observed. Commencement of blood circulation, that is the blood circulation within the embryo will start by this stage. Formation of optic cups, यानी कि जो eye का जो portion होता है, okay, and even the lens vesicles are formed by this time of the cheek embryo stage. Formation of visceral cleft can be seen. Development of auditory vesicles, okay, you can see जहाँ से उसे वो था ना sense, hearing sense, the auditory vesicles are developed in by this stage. There is a differentiation of 28 somites in the body. Now, since it is a 48-hour cheek embryo, you will see 28 somites in this particular cheek embryo. The development of extra embryonic membrane can be also be visualized by this stage. Moving and talking about the shape of the embryo, the 40-hour cheek embryo looks like a question mark. Okay, if you see it properly in the mirror view, it will give you a question mark like a sign. In this stage, the development, the head region shows prolific growth. Okay, mainly you can see the growth taking place in the head portion, that is the cranial portion and not the caudal portion. Because of this, because of which the embryo is getting this peculiar question mark shape. Because ये development एक जैसा नहीं है पूरे body में. Majorly by this time only the anterior part, that is the brain region, is getting developed. Talking about the flexures in the head region, you can see the bending which is taking place, okay, which we call as flexure. This bending is towards the right side and backward side, right and backward side ka bending hota hai. In 48-hour cheek embryo, two flexures are seen. Okay, one is in the mid brain region, because of which the fore and the hind brain become parallel to each other. You can see they are both running parallel to each other, and this flexure is called, because it is taking in the brain portion, it is called as cranial flexure. In the posterior region, you can see the brain, a cranial flexure is formed again. This flexure turns the head towards the right side of the body. This region will give the neck of the adult animal. यानी कि जो neck formation है, वो second flexure जो हो रहा है, cranial flexure, cervical flexure जो हो रहा है, उसके through उसको neck portion develop होता है उसके body में. Okay, this is again a mounting of a 48-hour cheek embryo, where you can see the brains properly, you can see the optic lens, the optic vesicles, you can see the heart which is getting very well developed, you can see the auditory vesicles present on the anterior part. You can see the somites are also laterally placed to the notochord. You can see properly in between the somites, you can see the neural tube which is running down to the posterior angle. Okay, talking more about the torsion in this animal, the 24-hour cheek embryo is very much flat, and because of torsion and flexure which is taking place, it is going to turn and twist the embryo and give it a little elevation. Okay, its ventral surface is in contact with the yolk still. As the development proceeds, the anterior end of the embryo will turn to the right side and will try and, it will still be connected on the left side to the yolk. Okay, the twisting of the body of the animal is determined as torsion. At the end of 48-hour cheek, the twisting will reach to the cervical flexure, and it is nearly 11th, at this stage, near the 11th, 13th somite, you can see the flexure taking place. Now, talking about the brain, as I've told you, the brain is divided into three portions, that is called as proencephalon, mesencephalon, and diencephalon. So the forebrain will show you two regions, that is telencephalon and diencephalon. The hindbrain will show emerge out as a medulla oblongata. Talking about the eyes, the optic cup is two-layered in this animal. The lens placade is started in the development. The process of the placade of the lens is started. Talking about the ears, the invagination of the optic vesicles is completed, and it is connected to the ectoderm of the by endolymphatic ducts. Talking about the elementary canal, the foregut reaches up to 1 by 2 millimeter in length. The heart is 5 prime shaped. Okay, 5 prime, if you see, it looks like a 5 prime shaped. It is tube like. अभी आप फिलहाल देखोगे तो ट्यूब लाइक दिखेगा आपको. It is not developed, it has not properly developed the chamber, but the formation of the chamber actually starts by the 48 hour. Talking about the blood vessel, as you see, there are the aortic, the two ventricle aortas are being formed, and the aortic arches are also starting to be developed, and the blood circulation is also increased. Talking about kidney in this animal, the kidney pronephros is completely developed. Mesonephric tubules have started to develop. Pronephric already developed हो चुका है. Now it is getting developed more into the mesonephric tubule structure. Somites in these animals of the 48 hour, the somites, the incubation is 25 pairs are seen, and the mesonephric tubule will develop from intermediate mesoderms. In this picture here, we can see the 72-hour cheek embryo. Okay, this one is much more developed. You can see much more bent, much there is more amount of torsion and flexion taking place in the animal, and the animal is more bent and going downwards. So at this stage, the area opeca and area pellucida will vanish away. It will not be visible to you. The extra embryonic area has grown in size. Okay, the size is growing. The extra embryonic area is growing in size. The primitive streak will disappear by this stage. The mesoderm in front of the Hense's node has given rise to 36 pairs of somites. The brain have differentiated into telencephalon, mesencephalon, metencephalon, and myelencephalon. The heart has been differentiated into ventricles and atrium. The eyes are differentiated into optic cups. You can see in this picture how well developed the optic cups can be seen, and the lenses and the optic vessels also have developed sufficiently. The head region, if you see, it has bent on the right side due to cranial flexion. Four pairs of gill clips have been developed in this animal. The cranial nerves and the ganglia are quickly developing, which with one new cranial ganglia forming in the body of this chick. The organ of digestive system is very well formed, or it is almost along with the midgut. Okay, so you can also see the bending and the clips, gill clips which are formed. The tail bird is greatly developed and has given rise to allantoic stalk and tail portion. So you can see the tail bud and the leg buds in the animal. In the posterior part, you can see here, these are the leg buds and this is the tail bud portion. The lateral amniotic fold, the vitelline artery, and the anterior omphallomesenteric veins have also been developed in these animals. In middle region, a pair of forelimb over here, you can see these are the forelimbs, bud are rising, and in front of the tail, a pair of hind limb buds are developed, which will rise and form the limbs later on. The olfactory pit or the visceral arches, amnion, allantois, and amniotic cavity are also developed. The cranial nerves and the ganglia are quickly developing, which with one new cranial ganglion with per neurosome. Okay, per the neurosome there is a ganglia which is forming in the body of this chick. The organ of digestive system is very well formed, or it is almost on the way of the formation. The organs of the digestive system are forming. At this stage, the embryo has a foregut, hindgut, and the beginning of organ also along with the midgut. Talking about the orgogeny, what is organogeny? That is formation of the organs is called as organogeny. Okay, just give me a minute. I have little problem with slide sharing out over here. Okay, in a while, just give me a minute. I will quickly share my screen back with you. Yeah, talking about organogeny, formation of organ is called as organogeny. Extra embryonic membrane or the fetal membrane or the yolk sac formation. The chief embryonic food is yolk, which is surrounded by a sac-like structure like the investing membrane, which is called as yolk sac. Yolk sac is made up of splanchocosphere. The function of the yolk sac is to protect the yolk and keeping it in the position it will help it to digest and absorb it. Yolk sac serves as a primary organ of nutrition to the embryo. Talking about the amnion, it is made up of inner ectoderm. The space between the amnion and the embryo is filled with amniotic cavity, and the amniotic cavity is filled with amniotic fluid. The function of this is to protect the embryo from any mechanical jerk और प्रिवेंट इट फ्रॉम डेसिकेशन, यानी कि अगर कहीं गिर जाता है अंडा तो अपने एम्ब्रियो को कोई प्रॉब्लम ना हो, उसे प्रोटेक्ट करने के लिए एक गैप रहता है, एंड इस गैप में एक एमनियोटिक फ्लूइड रहता है जिसमें एम्ब्रियो फ्लोट कर सकता है इजीली, ताकि उसे कोई डैमेज ना हो, एज वेल एज कोई डिहाइड्रेशन ना हो, पानी का कमी ना हो जिससे वो सूख के मर ना जाए, इसलिए अगेन इट प्रिवेंट्स द डेसिकेशन ऑफ दीज एम्ब्रियो. Then comes the chorion. Chorion, it is made up of outer ectoderm. It is the cavity between the amnion and the chorion is called as chorionic cavity. The function of it is to provide the liquid medium for the embryo. Then comes the allantois. Allantois, the bladder like structure. Okay, it is which is called as allantois. The mesodermal layer joined the allantois with the chorion called as allantochorion. A compound layer is formed. The function is it serves as a respiratory organ, excretory organ, as well as nutritional organ for the given animal. So that was all for today about the cheek embryology. The remaining part of the extra embryonic membrane we will study in our next lecture. Thank you everyone. Bye.